Hi everyone, this is my speciation overview video. So I've got some bits of paper with the key things on it and some little props to help us understand it. So this standard is about describing different patterns of evolution, and a real key to it is natural selection. And so I've got population of dinosaurs over here, and so we're going to start off with variation in our population. There will always be selection pressures on populations, which are things that will influence what is selected, what is advantageous and what isn't. So we're going to say that perhaps this dinosaur here with the big high frill on his back is that's not a very useful thing to have. It gets eaten more, noticed more by other predators, so that's not going to go on and survive and have as many babies. They're going to say the orange one's quite brightly coloured and also obvious to predators. But we'll say that these two are more camouflaged colours and so have an advantage. So the selection pressure might be predators seeing them. Favourable phenotypes are ones with more camouflaged colours, less easy to see. So those favourable phenotypes, they are more likely to survive, more likely to have more babies, and so they will then pass on the alleles for these favourable phenotypes. And evolution is basically the changing in these alleles that are passed on, the allele frequency, how that changes bit by bit over time. And it's the changes the allele frequencies in a gene pool that is evolution. And so it could be that this change will lead to just this species evolving over time. So it's already gone from being a population with lots of different colours, and now, so we're going to say it's still the same species, but they've got less colours. And over time they might become speckled green and brown or whatever. They're just going to stay the same species, still have got gene flow, but change with time. Or it may be that they change and become new species. So over even more time, it might be that the brown species, starts eating different things or living somewhere where brown is the best camouflage, and the green one might end up doing something a bit different, and they could end up being different species. But in both scenarios, allele frequencies in the gene pool have changed. Okay, so now we're going to look at evidence for evolution. Move that dinosaur. Okay, so I'm doing this at this point because it's something people find a bit hard, and they often will add it into a question. So there's lots of different kinds of evidence that you can use. There could be fossils, so there could be some bones found. They could be ancestral bones from a long time ago. If they're a fossil, they will be. They might be transitional fossils, so they might show changes of species that are sort of partway between one and another that we know about. But we can tell things about the timing, how long ago things lived from fossils with dating, and carbon dating, that is, and something about the structure of the species. Other structural things are comparing the anatomy of, it could be fossils or it could be current species. And this is where those terms homologous and analogous come in. So homologous structures, I think of homo meaning same, it's like a homosexual. So they are the same, they started the same, and they've become different over time. So homologous structures are ones that are evidence for divergent evolution. And then analogous are basically the other ones. So analogous features are ones that were different to start with, but over time they've developed similar function, and they are evidence for convergent evolution. Biogeography is when things, it's depending where they live, is the key to that they are linked in an evolutionary sense. So we've got this is the Earth with the one landmass that we started with, and then I know it's a long time ago to us, but as the land masses moved apart, those species ended up living in quite different places, like maybe Africa, Australia, they like to fall over, and Asia or something, maybe South America, Africa, and Asia somewhere. And you think, well, they're actually quite similar. We're going to say they're similar species. but living in really different places and different environments. So how are they similar? And they're similar because that they did used to all live near each other, and there was gene flow which kept them similar. And then even though it's a very, very long time that the Earth has moved apart, with species evolution it can sometimes take even longer for things to change. So it's basically where things live and how similar or different they are. Okay, then we have looking at DNA, and you can look at nuclear DNA, mitochondrial DNA, and Y chromosome DNA. Mitochondrial and Y chromosome DNA are useful from the point of view of timing because they have fairly regular mutation rates and they're not affected by the processes in meiosis. So you can look at changes through time with them. And with nuclear DNA, you can look at whole sequences of the DNA from one species and another species and compare them and see what similarities and differences they have. And then finally, protein. Some species share certain proteins they make and other species won't have those proteins, so you can look at whether they had common ancestors and that sort of thing. And that's all we'll say for evolution for now. So speciation, moving the fossils out the way. So we have, with natural selection and the changes over time in our gene pools, we can have the phenotypes. These are graphs of phenotypes, either changing to be more stable, so the feature, the sort of central in the bell curve is the most useful one. It might be directional. Over time the features might change to be more of a type at one extreme or the other extreme of the phenotype, or disruptive, where the middle one is not so useful and ones on the extreme are different, and this will be where you'd get two different species potentially, whereas this one will just be a change, evolution within a species, as with that one. Okay, then we have our two key types of speciation, allopatric and sympatric. I think of sim meaning same, and so these are ones that live in the same area, but for some reason gene flow is not occurring, whereas allopatric is a different geographic area. So for some reason something that was the same species all living in one area, I'll bring our dinosaurs back, they have ended up being separated for something, and so it's the geography that's keeping them apart and stopping gene flow between them. And whenever things are living in other different places, the environment's just going to be different. It's going to have different selection pressures, and so over time what the food and the predators in the environment like is here will be a little bit different there. These ones will have different mutations to those ones, and so over time they will just become more and more different until eventually they are so different that if you reintroduce them, they would no longer recognise each other as the same species. Sympatric's a little bit harder to understand because you think, well, how can it be if they're all in the same area that they end up becoming different? So it's not a geographical barrier this time. Something else has happened to change the gene flow. There might have been a mutation so their behaviour became different, or something to do with the ecology of the area or the niches. Like these ones might end up eating a slightly different food to these ones, and again over time those changes accumulate until they might be different species but living in the same area. And then polyploidy, I'll do some more on this at the end. That is an instant change in the number of chromosomes, the number of sets of chromosomes, and that will instantly make you a new species. And so that is a sort of subset example of sympatric because it happens in the same place. So how these changes build up over time and cause them not to be able to breed and have gene flow is our reproductive isolating mechanisms. And the example of this I'm going to use is people. So we're going to say this is you, and you're trying to find a date to go to the ball. If you live in completely different areas, so she's in America and this one's in England, it's going to be very hard for you to get together and have babies, right? You just live far apart. You might live in the same sort of area, but still not cross paths. So it might be that Superman goes to Sacred Heart College and you're at some girls' grammar, and so you still sort of live in Auckland, centralish Auckland, but you just don't end up really seeing each other very much. Temporal is timing, so it might be that you're awake all night while Superman's asleep, and then you go to sleep and Superman's out and about going, where's that wonderful person? But you just don't meet each other in terms of time. It could be a behavioural thing. So here we go, we can see, I think she's winking. So you might wink at Superman and Superman's going, what on earth is that? She just looks weird, she makes some strange face at me. I don't think I want to be with her. So some sort of behaviour or courtship activity that is, they just think, no. Mechanical is when the actual bits that are needed for the exchange of gametes don't fit together. And then gametic is the gametes, the sperm and the egg, can't actually fertilise. Then we go over to post-zygotic. So we have actually managed to do all those things. All these things have happened, and we've managed to actually fertilise our gametes. And so we have a hybrid. And sometimes what happens is you can have fertilisation of the egg, but then it won't grow past a certain number of cells. It grows a little bit, but then something goes wrong and it gets miscarried. It could be that you have fertilisation, gets born. So here's our little creature, a horse or something, gets born, but then this offspring, even though it's able to survive, it can't have more offspring. So that it's sterile. And then the last one is a sort of weird one, is you manage to get some of these, you manage to get some more of that generation, but then when they try to breed, they can't make any more. So the species sort of just dies out, it can't sustain itself. Right, patterns of evolution. Okay, so we're going to talk about 6 different types. So gradualism and punctuated equilibrium are really talking about the timing of when the evolution, the change in species is happening. Gradualism is gradual, and so you'll have graphs where there's, like, at certain intervals, there's little lines indicating different species coming off. So this is time going this way. And so there's not sort of such sharp changes. They're more gradual changes over time. Punctuated equilibrium tends to have more horizontal lines, because if this is time, it's like you've got same species, same species, same species, and then suddenly over this very tiny space of time, we've ended up with 2 new species. The same here, and up here we've diverged into 3 different species, but the horizontal line is indicating that it's happening over a very short time, those changes. And so that's what they will look like. The long lines is going, well, we're the same, we're the same, we're the same over a very long time, so it's called stasis. Okay, then we've got convergent, divergent, and coevolution. So convergent evolution is when 2 things are converging to become, they were different, they're becoming more similar. So this is not a very accurate example, but we're going to use our iguana here and our llama. Maybe I'll put them sideways like this so you can see them better, because they're very cute. So we're going to say that these were quite different species. Well, they are really, aren't they? But if it was an example of convergent evolution, over time they would become more similar. So maybe they're living in a similar habitat and they've got a same sort of predator attacking them. They might end up developing the same sorts of defences, or eating the same food, or having the same colouring or something like that, because of their niche. If they were divergent, so they're going to start similar and become different, they may have started off, we're just going to pretend that they're similar, and then over time they become more different. It might be that the iguana is getting smaller and living in the undergrowth and getting green and more camouflaged and spiky, whereas the llama might be coming in the open plains and becoming bigger and developing hooves and, I don't know, other sort of attacking mechanisms. Divergent radiation also has a little subset called adaptive radiation, which instead of just going to a few different species over time, you tend to have lots of species happening, and they tend to happen all at once. So you'll get bigger numbers of species, and again, see the time scale is quite short, usually because lots of niches have opened up for some reason. And so suddenly there's different places for the animals to go, and it might be that in certain places there's different food or different selection pressures, and those selection pressures act sort of quite quickly over a short amount of time to make differences in the population. Okay, co-evolution, or use our llama and our iguana again. So that's when each other are the selection pressures for the other species. So it might be that we're going to say that maybe llamas eat iguanas, which is not actually the case, but we'll pretend. So our llama might want to eat this iguana, so the iguana might develop, just by chance, it's not doing deliberately, it's going to say some of the iguanas might develop some spikes on their back, and so those ones might be, you know, the llama's less likely to want to eat the spiky thing, and so the ones with spikes will survive. Natural selection story, they'll go on and have more babies, so that population will end up spikier. The llama might then develop the ability to avoid the spikes, and some of them might end up just naturally having some behavior where they eat the underside of the iguana, and so the spikes are not so useful anymore. Some iguanas might get a mutation where they develop a poison inside them, and so those ones will survive and live longer. And then the llama might, some of them might develop the ability to digest that poison, and so those ones will be the ones that survive. And so each change in the other one leads to a change in the other species, so how they evolve is in a relationship with the other species. And finally, we're going to quickly do polyploidy. Sorry this video is so long. So polyploidy is basically just two sets of chromosomes, more than two sets, sorry. 3, 4, 5, 6. And a set of chromosomes is how many different chromosomes, how many of each kind of chromosome you have. So humans are N number, which is the number of one of these sets. We have 23. And normal cells are diploid 2N, and so we have 46. We have 2 copies of all of our 23 chromosomes. But it could be that there's a plant and their N number might only be 4, compared with our 3. They have 4 different chromosomes. They might be triploid, so have 3 lots of their 4 chromosomes, so they've got 3 copies of each one, so they have 12 chromosomes in their normal cells. They might be hexaploid and have 6 copies of their set of 4, which they'd have 24 chromosomes in their cells. And you need to be able to talk about how we get polyploidy, what happens with the subsequent fertility, and the advantages of being polyploid. Don't get worried about too many of the huge range of terms that are in this, just get the basics in your head first. So it happens because of non-disjunction, and I'm going to try and show that here with some chromosomes. So this is a cell, and we've got two sets of chromosomes here with the homologous pairs, and so they get replicated, don't they, in meiosis. And so we have our copies, and then normally they'd line up at the equator, and these two would go over here, and those ones would go over there. But you could have non-disjunction at this point where the spindle fibers don't pull those ones that way, and all of them end up in this cell. So you get twice as many, this is quite hard to do, sorry, twice as many over here, and none in this cell. And then you do the next stage of division, and you're going to end up with twice as many in the gametes as you would normally have. And that's the key. You can get the disjunction done here as well, so you have a normal split to start with, but then the second splitting doesn't happen. Either way, you're going to end up with twice as many in the gametes as you should have, and so that will instantly be a new species. And depending how many you end up in the gametes and what they try to subsequently fertilize with, the fertility will depend on whether the number's even or not. If you have an uneven number, so 3, 5, 7, 9, whatever, as your sets of chromosomes. So say this has become a 2N gamete, and it might join together with a 1N gamete. You would have a 3N individual. That 3 can't split into 1.5, if that makes sense. So here we'll say that this is my 3. I'm a 3N, a triploid individual. When I go to do meiosis, I can't split that evenly into my 2 cells. I can't really do 1.5 chromosomes, and so that's why they're infertile. Any uneven numbered individual will be infertile. There's lots of advantages to being polyploid. The plants tend to be more vigorous, they have bigger fruit, they're stronger, more resistant to disease. The other thing with polyploidy that sort of might help it make sense a bit is that with plants, of course, the growing tip is what grows into the flower, and the flower is where the gametes are. And so you can also get non-disjunction in mitosis, in the cells are going to become the ovules, or the produce the pollen in the flower. So it can happen in mitosis as well because of how plants do reproduction. And I'm going to stop there because that's, crikey, 20 minutes. I'm sorry about the length of that. Hopefully some of it was fun. Feel free to use little things around your house to help explain it to yourself or to people in your family. Best of luck.